All right. Well, how different are you from the person who is no longer sitting next to you since you are at home learning remotely? Well, interesting, you are only 0.5% unique from that person. You are 99.5% identical. And so with that in mind, you say, well, why is this important? With that in mind, in order for you to be 0.5% of you to be unique, it means that you must have a tremendous amount of DNA inside your cells. And that's the truth. So a lot of our genes and a lot of our information is the same. We have information for eyes, we've got information for ears, we've got information for arms. We all have that same information. But within that information, there are small uniquenesses about us, only 0.5%. So again, if it's only 0.5%, there must be a huge amount because that would make that 0.5% a very large number. So some of this might be review from high school, from your last biology class. I think you might remember the term chromosome. So a long strand of DNA is called a chromosome. A gene is a small bit of information that is on the chromosomes. So genes are like the recipes. And when you look at our chromosomes, there are many recipes on our DNA, and each individual recipe is called a gene. So it's a sequence of the DNA that will code for an actual protein. That's what DNA codes for, is protein. It also codes for RNA. So both protein and RNA are the instructions for how to make them are encoded in our chromosome information, our DNA information. Now DNA, you're familiar with DNA. It's the A's, the T's, the G's, the C's. So it's those nucleotides, those 4 nucleotides. But when you look inside the cell, the DNA is actually wound up around a protein called histone protein, and that's what these balls here represent. That would be the histone protein that the DNA is wound up around. So if you were to open up the nucleus of a cell, you would find both DNA and protein. And so when those two are together in the cell, which is normally how they occur, that is called chromatin. Just a slight variation in terminology, but that means DNA plus that histone protein that they are wrapped around. Okay, so we have 46 chromosomes in each of our cells, as long as they're our body cells and not our gametes. So we're working on body cells right now. This number will change when we start talking about reproduction and the formation of gametes. But when you look at our body cells have 46 chromosomes. Those chromosomes come in pair. You get a chromosome number 1 from your mom. It's pair number 1 from your dad. You have a chromosome number 2 from your mom, a number 2 from your dad. Those are called homologous pairs. So they're coming in pairs. And if you look at the diagram here, where it's showing you the chromosomes there, it's actually showing you a chromosome that has been replicated. So, and the reason why we keep showing you chromosomes that have been replicated is because when they've replicated themselves, they're thick and chunky and they're condensed enough that we can see them. For the most part, when they're loose, like they are in this diagram up here, you and I cannot tell one chromosome from another chromosome. And so, in fact, when our cells go to divide and they make a complete copy of their DNA, the DNA has to become condensed and it gets thick and chunky, and now we can actually see it. And so what you're looking at here is actually one chromosome that has made a complete copy of itself, and its complete copy is identical. So even though that is actually two chromosomes right there, they are identical chromosomes. So our chromosomes come in pairs. You get a number 1 from your mom, a number 1 from your dad. And we know which pairs make up chromosome number 1 because they're both going to be the longest chromosomes inside your cells. It goes from number 1 all the way down to chromosome pair number 23. And pair number 23 are your sex chromosomes. And the ordering, the number ordering goes from longest all the way down to the shortest of these chromosomes. Our sex chromosomes are, they kind of are not included in all of that because if you are a female, you have 2 X chromosomes, and X chromosomes are fairly large. If you are male, you have an X and a Y, and the Y chromosomes are fairly small. So when we start talking about chromosomes, we talk about autosomes and sex chromosomes. So the first 22 pair are called autosomes, auto meaning body, and they're going to code for, your physical body features and all your internal features that we can't even see. Your sex chromosomes determine your gender, and so they're considered sex chromosomes, and they kind of are on their own set of rules. So anyway, those are our chromosomes, and we like to pair them up when we look at them like this. We pair them up to make sure that the individual has both members of the pair. In other words, you have a number 1 from your mom and a number 1 from your dad. You've got a number 2 from your mom and a number 2 from your dad. And of course, if we pair up the chromosomes, we'll get all the way down to the sex chromosomes. And if we do that, we can actually tell the gender of a baby, which is oftentimes why we would even be doing this work, is because not to determine the gender of the baby, but to make sure that from a genetic standpoint that the baby has all of the chromosomes that are necessary. Not too few, not too many. So here is a chromosome, and again, right here is 1 chromosome. But this chromosome has made a complete copy of itself. And the reason why it's duplicated itself is because it's received signals that lets the cell know that it's going to divide. So they become tightly packed in this process and of division. They duplicate themselves. So now I actually have a duplicated chromosome, and so these duplicated chromosomes are referred to as sister chromatids, because what's true about the sister chromatids is they are genetically identical to each other. So I'm just making an exact copy of myself. So when you look at it, here's some terminology, and this is your homework sheet as you recognize these terms, that these are the sister chromatids. They're exactly identical to each other. This one here is identical to this one here. They're held together in a region that's called the centromere. And then the genes are located on the chromosomes. So remember, that's just a segment of the DNA that codes for a protein or for RNA. And that gene right there is identical to whatever, to this one here. So if this is the gene for, let's say, brown eyes, then on the sister chromatid is the same gene for brown eyes. They are identical to each other. Okay, so 5.3 is getting us into this idea of the cell cycle. So we want to get to division and look at mitosis and look at meiosis, but we need to really understand where does that take place and what's going on in the typical life of a cell. So if you look at the typical life of the cell, then it goes through what's called it goes through a cell cycle, and all of your cells have a regular cell cycle, meaning that it's all timed. The way that this is going is all timed for growth and division. And as long as your cells are healthy, then they're going to be doing this at irregular intervals, very predictable. So unhealthy cells can change. They can do this in an unregulated fashion, and that's what cancer cells are. They're a cell that have lost control over the cell cycle. So if we look at the cell cycle is broken up into what's called interphase and the mitotic phase. And so interphase is the longest phase of the cell cycle. It accounts for 90% of the cell's lifetime, and it's where normal cell function is taking place. So all that stuff that we've talked about so far, as far as cellular respiration, photosynthesis, all of those typical cellular processes, you producing digestive enzymes, all of that takes place in interphase. So that takes 90% of the cell's lifetime, and it's where you're doing normal cell function. But then the cell receives a signal. Say you've injured yourself, you just stubbed your toe. When that signal is received by the cell then moves out of interphase into the mitotic phase. So the diagram looks like this, and so we're starting here. Here is our cell, and the cell is going to go about doing its normal cellular activity. So if it's a stomach cell, it's producing for you digestive enzymes, just normal cellular activity. If the cell receives a signal, like it's been injured, then it will, when it receives that signal, it will replicate its DNA. Once it's replicated its DNA, it's destined to go through the M phase or the mitotic phase. So notice that in interphase, cells doing its normal cellular activity, it receives a signal that, I need to make a copy of myself, so it replicates its chromosomes. So now it has twice as much, and that's where our sister chromatids now become evident. So here's where the sister chromatids are produced. Okay, so it receives a signal, and it makes a complete copy of itself. When it does that, the cell then is going to prepare for division, and here you can see a cell here that's going to divide. The cell's going to split itself down the middle. Those two splitting down the middle is going to require for them to divide the cytoplasm. And so the splitting of these two cells down the middle is a process called cytokinesis. So you divide your DNA information, you divide the cytoplasm. So we would say that during the mitotic phase, right in here, mitosis literally means the division of the DNA or the chromosomes, and then cytokinesis is the division of the cytoplasm. And when you're done, you will have two offspring cells that are genetically identical to each other. Okay, and it looks like this. So this is what we would be seeing in our lab. Actually, it looks like this. So this is what we would see in the lab under the microscope. So there are 4 phases to mitosis. There's prophase, metaphase, anaphase, and telophase. So prophase is where the chromosomes become condensed and you can see them and the nuclear membrane breaks down. Metaphase, the chromosomes line up in the middle. Anaphase, they're pulled apart. And telophase, they end up migrated on separate sides of the cell, and now the cell is going to split